हेलो। अरे अब हेलो।
Good afternoon, everyone. It is my honor and privilege to welcome you all to today's special session of the Translational Lecture Series, a platform that celebrates knowledge sharing across disciplines, fostering new perspective and innovation. To commence today's program, may I request the dignitaries to kindly occupy the dais. Director CDRI, ma'am, Dr. Satinath, Dr. Biswajanani, Dr. Arun Trivedi. Thank you, esteemed guests. Now I'd like to request Director CDRI to kindly extend a floral welcome to Dr. Satinath and Chief Guest Dr. Biswajanani as a token of our appreciation and respect. Thank you, Director CDRI, ma'am. Next, may I request Director CDRI to develop the welcome address and offer the opening remarks for this session of the Translational Lecture Series. Director.
Thank you, Baisakhi, and good afternoon, colleagues, online participants, and my dear students. I welcome you all to today's program in the Translational Research Series. This is also part of our 75th year commemorative activities. So we're bringing two things together in one. As you know, this particular lecture series is aimed at bringing clinician researchers to CDRI, so we learn about their work, their perspectives, and their ideas on unmet needs that institutions like ours should be working on. Our last talk in the series was in May, when you recall Dr. Anurag Agarwal was here, and he joined us in the series. So, I say this time and time again, why do we need a lecture series like this? Well, it's clear that all too often we fall into silos and we all work in our own comfort zones and don't look at how our work has relevance in the broader context. For an institution like ours, which does drug discovery and development work, it is very, very, very important that we really understand the unmet clinical need from a clinician perspective. Just to give you a sense of how engaged we are with clinicians, something like only 10% of our proposals have a clinician as a co-PI. And I think we can really work towards improving that ratio significantly, so that our proposals and our research is more aligned with what the unmet need is and differentiated from what is already available in the clinic. So I'm really, really delighted to welcome today's speaker, Dr. Satinath Mukhopadhyay, former professor and head of the Department of Endocrinology at the Institute of Postgraduate Medical Education and Research, Kolkata. He's a well-known physician with over 35 years of experience in patient care and teaching. Equally, he has been a prolific researcher. His areas of interest include understanding insulin resistance, bone metabolism, diabetic neuropathy, PCOS, and other diabetes-related conditions from a mechanistic perspective. His work spans the whole gamut, from cohort-based observational studies to clinical trials for drugs to testing disease biomarkers. So we're very fortunate indeed to have such a remarkable clinician researcher in our midst today. Thank you for accepting our invitation. And although this is your first visit to CDRI, we hope that there will be many more. I also want to welcome today to this program Dr. Vishwajanani Satgiri, Head Traditional Knowledge Digital Library Unit at CSIR. Dr. Vishwajanani is, of course, no stranger to CDRI. She's a member of our Research Council. She's an organic chemist by training. She has a PhD from ISC, and she has experience both in academia and in industry. Now, she's put her heart and soul into creating a usable, accessible database of Indian systems of medicine, which include Ayurveda, Siddha, Unani, Saurikpa, as well as yoga. And this is mainly done from the perspective of patent offices, so that traditional knowledge is known in the prior art space and that no undue patenting happens and there is no misuse of traditional knowledge. So to date, more than 4.5 lakh formulations and practices have been transcribed into the TKDL database, which is, as you can imagine, not just a formidable achievement, but also something with great value. And hopefully, as it opens up to researchers and others, this digital library will be the starting point for a lot more research. So thank you for joining us today. Before I hand the podium back to Baisakhi, I want to thank the organizers of today's event, Dr. Arun Trivedi, who heads the Translational Research Series Committee, and the committee members, Dr. Vivek Bhosle, Dr. Baisakhi Morana, and Dr. Rajdeep Guha. So with that, I'll hand it back to Baisakhi, and thank you very much for being here.
Thank you, ma'am, for your encouraging words and vision. Now it is my privilege to introduce today's distinguished speaker, Dr. Satinath Mukhopadhyay. Dr. Mukhopadhyay is a highly respected endocrinologist with an MD and DM in endocrinology. He is the former professor and head of the Department of Endocrinology and Metabolism at IPG-MER, Kolkata. His contribution to the field span decades and have a profound impact on both clinical practice and academic research. He holds prestigious fellowship from Royal College of Physicians, London, and is a fellow of the National Academy of Medical Science as well as West Bengal Academy of Science and Technology. Dr. Mukhopadhyay has an outstanding publication record with over 300 scientific publications, more than 20 book chapters to his name. He has an H-index of near 50 and has received over 60,000 citations, reflecting the wide-reaching influence of his work. He serves as an associate editor for Diabetic Medicine and Acta Diabetologica and is the editor-in-chief of Metabolic Syndrome published by Elsevier Global. His remarkable achievements has been recognized globally, including WIDF Outstanding Achievement Award from the Mayo Clinic, USA, and an invited lecture at the American Endocrine Society in 2023. Dr. Mukhopadhyay has received multiple oration and honor over the years. His key research interests include insulin resistance, metabolic bone disease, and diabetic neuropathy, areas that are critical in understanding and management of diabetes and metabolic disorders. We are honored to have him with us today. Please join me in welcoming Dr. Satinath Mukhopadhyay to deliver his lecture.
Good afternoon, everyone. At the outset, I would like to thank Dr. Radha Rangarajan, Director CDRI, and Dr. Satigeri, Dr. Arun Trivedi. And I would also like to put it on record that Dr. Rajdeep was particularly instrumental in acting as a bridge between CDRI and myself for having me over here. It's only in the fitness of things that we are having this talk on 28th of July when insulin was discovered. And we'll be speaking on insulino-mimetic and insulin sensitizing agents today. So insulin was discovered back in 1921. There are some controversies as to the exact date, but on record, this is 20th July, and Diabetes Association of India, which started off from Calcutta, we continue to celebrate 20th of July as the Insulin Discovery Day, right? So I have no disclosures related to this presentation. So what are the unmet needs for treatment of diabetes in India? If you look at type 2 diabetes, which is believed to result predominantly from insulin resistance, as far as Indians with type 2 diabetes is concerned, we have a double whammy. We have extremes of insulin resistance coupled with extremes of pancreatic beta cell dysfunction. Maybe one of the reasons why type 2 diabetes manifests a decade earlier in India compared to that in the Western world. And Indians with type 2 diabetes also need greater use of insulinotropic agents. I mean, the agents that stimulate insulin secretion and synthesis from the dysfunctional pancreatic beta cells. Now, incretin-based therapies are the in thing now in all the modern treatment algorithms of type 2 diabetes. And of them, we have two kinds. One is the DPP-4 inhibitors, which are cheap, readily available, but with not so good anti-hyperglycemic potential when used as a monotherapy. On the other hand, GLP-1 agonists or the dual agonists, they are powerful stimulator of pancreatic beta cell dysfunction, with the added advantage that they stimulate the secretion of insulin in a glucose-dependent manner. So when used as a monotherapy, they do not produce symptomatic or biochemical hypoglycemia. But that said, the majority of our T2DM population cannot access GLP-1 based therapy. The monthly cost of treatment with tirzepatide, which is the dual incretin agonist, is around 17,000 rupees in a modest dose. Talking about injectable semaglutide, it has been launched last week only in India, and the cost is supposed to be around 24,000 rupees a month. And hardly having any insurance coverage, how many of our patients can really afford to go for GLP-1 based therapy? We know pioglitazone nicely addresses insulin resistance, and there are data to suggest that it may be more effective in Indian patients than their Western counterparts. We do have such data from India, but the drug is associated with multiple side effects. So there is a clear unmet need for effective, safe, and affordable anti-hyperglycemic molecules in the treatment of type 2 diabetes. The problem becomes even more important if you go to type 1 diabetes. We keep saying that type 1 constitutes less than 5% of all diabetic people in India. But considering the sheer number of Indians, if you talk in absolute terms, we have more than 100,000 children and adults with type 1 diabetes in India today. They're essentially dependent, their survival is dependent on insulin, and insulin therapy remains a challenge. If I may refer you to this extremely sobering story which came out in Times of India last week, a father committing suicide for not being able to afford or buy insulin for his daughter with type 1 diabetes. So intensive insulin therapy has logistic challenges, it's expensive, and it needs to be monitored. How do you monitor glucose levels? Glucometers are simple, but they do not give you the time that you spend in the hyperglycemic range or the time in range, which comes out with continuous glucose monitoring systems. The systems currently in use in India, they last for around 2 weeks and cost 6,000 rupees. So just for monitoring the type 1 diabetic, the parents need to spend 12,000 rupees a month, leave alone the cost of insulin and the devices used for injecting insulin. So forget about insulin pump, which I put there. In the whole of Eastern India, we have around 400 patients with insulin pump. The pumps cost anything between 5,75,000 to 6 lakh rupees. And the pump needs to be attached to your glucose monitoring system. So CGM is an essential component of the pump, right? So it's frightfully expensive. The majority of our people cannot afford this. So these are the unmet needs, and I put a question at the bottom. So vanadium, being known as both an insulino-mimetic agent and an insulin sensitizer, can it fill up some of the voids that is there in the treatment of both type 1 and type 2 diabetes in India? And at least in type 1 diabetes, can it serve as an insulin sparing agent? Instead of multiple subcutaneous injection, we put the patient on a basal insulin to take care of their pre-meal and fasting glucose levels and take care of the post-meal rises with, instead of multiple doses of bolus insulins. Could vanadium have a role? So vanadium, this is very interesting. Yesterday when I was arranging my talks, I just wanted to see what AI has to say about vanadium. So I asked Meta AI, what's the current status of vanadium in diabetes, and it came up with the following bullets. So it's a trace mineral present in the diet. Common dietary sources are mushrooms, black pepper, parsley grains, and drinking water. A typical diet has 10 to 60 micrograms of vanadium depending on the type of food that you have. And the AI did not forget to mention that vanadium might act like insulin or help to increase the effects of insulin. So it's echoing our thoughts actually. Insulino-mimetic and insulin sensitizing effect. And it also told me that currently, if you Google for vanadium preparations, you can access 27 brands over the counter. And these are being used by athletes, obese people, people with type 2 diabetes, pre-diabetes, cancer, and so on. But there is no good scientific evidence to support these uses. So what we need to generate are scientific evidences to back up the claims made by people like me, or maybe many others, about the effects of vanadium, efficacy and safety of vanadium. Interestingly, this is a Chinese paper where they look at the plasma vanadium concentration in a cohort of people with newly diagnosed type 2 diabetes. What they found was that participants in the highest quartile of plasma vanadium had a notably lower risk of new onset type 2 diabetes. So vanadium kind of protected against the development of type 2 diabetes compared with those in the lowest quartile. And this was published in the American Journal of Epidemiology, as you can see. No such Indian data exists. Then this paper is from this city itself. So this is from Integral University, Lucknow. So, and that's from the chemistry department. They tell us about how to have effective shift basis of vanadium to make the molecule clinically available, bioavailable. Then I came across this. If we see vanadium partnering with insulin, which is what I was alluding to a little while ago. Can vanadium combine with insulin to have an insulin sparing effect? So in this paper, they identified the mechanistic pathways where vanadium works on the insulin post-receptor cascade. They basically identified 3 nodes. One is insulin receptor phosphorylation area. Next is the lipid handling by the adipose tissue. And third is the mitochondrial dysfunction. So these are the 3 areas that they highlighted. Busy slide. So nodes 1, 2, and 3 are identified here. You can see node 1 is IR phosphorylation. So how does vanadium maintain phospho-IR concentrations in the post-receptor pathway? Actually, the main mechanism of action of vanadium is believed to be inhibition of phosphotyrosine phosphatases. And when PTPs are inhibited, your phosphotyrosine remains in enough concentration within the cell to stimulate the downstream pathways. Node 2 focuses on ACT PKB phosphorylation. Again, vanadium maintains phospho-ACT and phospho-PKB, and when that happens, you can have downstream effect on the adipose tissue. Very briefly, if you see, the cartoon tells us that it promotes lipogenesis, it inhibits lipolysis, and allows for safe storage of fat in the adipose tissue. These are exactly the mechanisms being claimed by Eli Lilly for their tirzepatide. Tirzepatide is a GLP-1 GIP combo, and Lilly tells us that our competitor Novo's semaglutide does not have the GIP-1, so semaglutide does not have any effect on adipose tissue. It's only GLP-1 which you already have. It's the GIP that allows for safe storage of fat in the adipose tissue, inhibits lipolysis. Exactly similar mechanisms have been published with vanadium. Problem is, Lilly did not publish so far any data to show how GIP in the adipose tissue causes safe storage of fat and inhibits lipolysis. It's not known. And GIP alone, people in the past tried to use as an anti-diabetic agent because it's one of the incretins. Type 2 diabetes is characterized by a state of GLP-1 deficiency and GIP resistance. So GIP alone did not work. In fact, in animal models, GIP has been shown to be an obesogenic agent. And if you knock out GIP gene, obesity gets ameliorated. Type 2 diabetes goes into remission. GIP antagonists have been used to treat type 2 diabetes. Whereas here we are saying, when you piggyback GLP-1 on GIP, tirzepatide, actually the base molecule is GIP on which GLP-1 is piggybacked. So the entire thing changes. GIP becomes metabolic friendly and does all the good things that pure GLP-1 agonists lack. So that's what they claim. But there are no mechanistic studies as to the exact effect of GIP on adipose tissue. Very interesting. I told you insulin was discovered in 1921, and Elliot P. Jocelyn, those of you who have visited Boston may have seen Jocelyn Diabetes Center, a premier research center for diabetes in the US. So the founder, Elliot Jocelyn, back in 1923, just remember 2 years after the discovery of insulin, when the concept of diabetes was absolute insulin deficiency, no real concept of metabolic syndrome, he had the foresight, he had the vision to say that from an excess of fat, diabetes begins. and from an excess of fat, diabetics die. Before the discovery of insulin, starvation therapy was considered as a major treatment for treatment of type 1 diabetes also. Those patients actually survived on starvation. Others died within a week, and those who were forced to starve died a month later. So lifespan was prolonged by around 3 weeks with starvation treatment. We all know about this. You can have two kinds of adipose tissue, metabolically healthy, metabolically unhealthy. In response to a surfeit of nutrients, healthy white adipose tissue expands by hyperplasia. The cells divide. When you have a hyperplastic adipose tissue, the vasculature keeps pace with the rate at which hyperplasia occurs. So there is no tissue hypoxia when you have adipose tissue hyperplasia. Since there is no hypoxia, there is no hypoxia-induced fibrosis and inflammation. So you have infiltration with anti-inflammatory M2 macrophages and T regulatory cells, and you have formation of new vasculature. So this is considered healthy white adipose tissue expansion. As opposed to that, when you have unhealthy white adipose tissue expansion, it occurs through hypertrophy and not hyperplasia. When you have hypertrophy, our physiology teacher used to tell us, when you have hypertrophic adipose tissue expansion, the adipose tissue expands in cubes, but the vascularity expands in squares. So there is a deficit. And that's what leads to adipose tissue hypoxia and invites inflammation that eventually leads to fibrosis. And you can see infiltration with pro-inflammatory M1 adipose tissue macrophages and the natural killer cells. Angiogenesis goes down, and then you have metabolic syndrome setting in. So adipose tissue is the seat of chronic low-grade inflammation in obesity that fuels insulin resistance, and this is a recent coinage. Inflammation leading to metabolic syndrome. By inflammation here we mean chronic low-grade inflammation, and not the kind of inflammation you see in acute infection, in typhoid fever. Low levels of CRP just above the upper limit of normal, chronic low-grade inflammation, a lot of inflammatory cytokines in the circulation, and the levels of anti-inflammatory cytokines going down. So that is what is known as metaflammation. So in this paper in diabetes, we actually discussed what happens when you have hypertrophic adipocytes in abundance. They secrete a chemokine called chimerin. Chimerin inducts and binds to its cognate receptor on the plasmacytoidendritic cells. By a different mechanism which goes in parallel, extracellular nucleic acids, upon release from the inflamed adipose tissue, they bind to their, the high molecular group binding protein 1, which is their bona fide binding protein, and the combination goes and binds to the receptor for age, which is for the students advanced glyceridine products. So age-range interaction. So here you have age-range interaction from the extracellular nucleic acids on the one hand, and stimulation of TLR9 by chimerin binding to its receptor. TLR9 then goes on to stimulate type 1 interferon that causes macrophage polarization, and as you all know, polarized macrophages release a lot of pro-inflammatory cytokines that eventually leads to insulin resistance by mechanisms which I will discuss in a moment from now. So that's the paper, adipose recruitment and activation of plasmacytoidendritic cells cause metaflammation. The paper was well received by the Indian print media, as you can see. They were all praised for this, no harsh criticisms here. And then there were multiple publications looking at the relationship between metabolic syndrome and chimerin. Of interest here is this one, as you can see. Both insulin and metformin regulate circulating as well as adipose tissue chimerin. And metformin has been shown to reduce chimerin levels both in the circulation as well as in the adipose tissue, and insulin does the same thing. Now, I will walk you through papers a little while from now, which will tell us that we now have compounds in phase 2 clinical trials. They are a combo of metformin and vanadium, metforminium vanadate, and some similar other compounds. So they have been shown to ameliorate insulin resistance in metabolic syndrome. So metformin is already established. If you combine it with vanadium, maybe you get an enhanced effect on the insulin resistance pathway. Now, with these papers, we wanted to see if chimerin could be used as a biomarker to track people with pre-diabetes or type 2 diabetes. Now, as you all know, type 2 diabetes is a heterogeneous disease, affects mostly the elderly, 50% asymptomatic, associated many a time with multiple comorbidities. So in order to fulfill our research objective, we wanted to have a cohort of kind of pure pre-diabetes or type 2 diabetes of relatively recent onset with not much of comorbidities. So we thought, why not take PCOS as a model? You know, PCOS, the young girls, they have a very high risk of not only type 2 diabetes but also cardiovascular disease at a very young age. They are 3rd, 4th, 5th decades. They are not protected by the effects of estrogen, as it is believed to protect against cardiovascular disease in premenopausal women. When you have PCOS, that protection seems to be gone, it's lost. So we included a cohort of PCOS subjects and stratified them according to their glucose tolerance. We gave them glucose challenge, and so they are classified as normal glucose tolerance, pre-diabetes and type 2 diabetes. Surprisingly, 20% of patients, young girls between 18 and 25, they did not know that they had diabetes. On glucose challenge, they turned out to be having frank type 2 diabetes. And then we measured their chimerin levels. Serum chimerin levels correlate with the severity of dysglycemia in young adult women with polycystic ovary syndrome. And if you look at the data here, the chimerin levels are at the bottom. You can see second column, PCOS normal glucose tolerance 181, PCOS IGT 360, PCOS with type 2 diabetes 479. So there is a clear separation, an incremental chimerin values when you progress from normal glucose tolerance to IGT to type 2 diabetes. And if you look at the correlation between chimerin levels and body fat, the second line, you can see there body fat percentage. So 32 BMI, 38 BMI. And for Indians, any BMI above than, that is the body fat percentage, with not much difference in BMI. And BMI has now been recognized to reflect obesity status poorly in South Asians. This has been recognized by WHO also. So they ask us to shift away from BMI and go for body fat estimation. And for Indian girls, 30% is the upper limit of total body fat percentage. And here you can see 38%. Pre-diabetes 33%. So huge amount of body fat associated with truncal obesity, and that leads to, with high chimerin, so leads to insulin resistance. So this shows the correlation between body fat percentage and chimerin. Triglyceride to HDL cholesterol is now considered, the ratio is now considered as an independent predictor of cardiovascular risk in metabolic syndrome. And so here is the correlation between TG to HDL-C ratio and your chimerin level. And like I said before, you need to have pancreatic beta cell dysfunction in order to develop type 2 diabetes, no matter how insulin resistant you are. And this is very important in our endocrine practice, where we see children coming to us with syndromes of severe insulin resistance. They have insulin levels in their hundreds, sometimes even thousands. They have florid clinical markers of insulin resistance, acanthosis nigricans and skin tags. They never develop type 2 diabetes. So why is that so? Because their pancreatic beta cells are able to compensate for the prevailing level of insulin resistance. So the beta cells are robust, they work over time to produce hyperinsulinemia to the extent that those young kids, they do not develop diabetes. So insulin resistance is a necessary but not a sufficient condition to develop type 2 diabetes. In order to have type 2 diabetes, you must have pancreatic beta cell dysfunction, and here is the relationship between K-merin level and your HOMA2 beta, which reflects pancreatic beta cell dysfunction. So higher the K-merin level, higher the pancreatic beta cell dysfunction. And that's why these young kids with PCOS, they go on to develop type 2 diabetes and pre-diabetes at such a young age. Then we did this ROC curve and came up with the optimum trade-off between sensitivity and specificity for type 2 diabetes. You can see 85%, 89% sensitivity and specificity. It's not so good for IGT, but at around 80% sensitivity and specificity to pick up type 2 diabetes. Now, if you compare this for the sensitivity and specificity of an oral glucose tolerance test to pick up type 2 diabetes, K-merin performs better. So that's why we propose that this may be taken as a biomarker to screen community with type 2 diabetes. Young girls, PCOS, do not know about type 2 diabetes at this point of time, but maybe at least in that subset. Now, shifting gear from here, type 2 diabetes is an interplay between multiple organs, mainly liver, adipose tissue, skeletal muscle, pancreatic beta cells. And now cardiomyocytes are also being featured as a component of metabolic syndrome. So the most important interplay appears to be between liver and adipose tissue. So there is some kind of a crosstalk going on. As far as adipose tissue is concerned, we now have K-merin as a biomarker. There are several others, but we have picked up K-merin and we have shown its efficacy, sensitivity, specificity. What about the liver? When you load liver with fat, so what you end up is known as your non-alcoholic fatty liver disease, or the recent terminology metabolic dysfunction associated steatotic liver disease. Why the terminology got changed, maybe we can discuss during the Q&A. It will take quite a lot of time. But both ferritin A is the most powerful hepatokine in terms of insulin resistance. And when you have fat in the liver, higher the fat content, greater is the output of ferritin A from the liver. This has been very well characterized. So K-merin and ferritin A are both implicated in metabolic disorders like obesity and type 2 diabetes. The levels are often studied in relation to these conditions, and both K-merin and ferritin A levels are elevated in individuals with MASLD. So ferritin A is a hepatosecretory protein. CQ27 is the location of the gene. Very interestingly, adiponectin gene is also located on the same chromosome. So whether there is any relationship between the two, because they go in opposite direction. Wherever you have a low adiponectin, you have a high ferritin A. So we measured ferritin A concentration in people with pre-diabetes. We published that in Diabetic Medicine. And so pre-diabetics with high ferritin A level, in the highest quartile, they had higher BMI, the interleukin 1 beta, and more severe NAFLD. In those days, the nomenclature was NAFLD. It has now changed to MASLD. High ferritin A levels in pre-diabetes were associated with a higher risk of progression to type 2 diabetes. compared to those who did not progress. We followed a cohort for 3 years. In our cohort, as well as in the South Indian cohort reported by Dr. Ramachandran, we have the one, as Indians, we have the highest risk of progression to type 2 diabetes from pre-diabetes. 18% in Ramachandran series, 17% in our cohort. If you compare this to the Caucasian database, 2 to 5% is the rate of progression from pre-diabetes to type 2 diabetes. Chinese have reported 11%. Here it is 17-18%. So in 3 years' time, most of those who were supposed to convert to type 2 diabetes would have converted to type 2 diabetes, and we found that these Phe2 A levels predicted who would progress to diabetes and who would not. Now, in the middle, you see a zoomed in view of a lipid-loaded hepatocyte. So this is the fatty liver you can make out from here. You can see that the liver is loaded with fat. So Phe2 A released from lipid-loaded hepatocyte causes insulin resistance basically by 2 mechanisms. The first is that Phe2 A is a powerful inhibitor, direct inhibitor of insulin receptor tyrosine kinase. So that's one. And the maximum effect is seen in skeletal muscle, because 70% of glucose uptake in skeletal muscle following a meal, glucose uptake in following a meal, 70% of it occurs in the skeletal muscle. So it's inhibited, explaining the rise in postprandial glucose levels. And Phe2 A binds to toll-like receptor 4 present on the surface of inflamed adipocytes and immunocytes. When that happens, you have downstream production of inflammatory cytokines that give rise to insulin resistance. So here in this paper, we show how palmitate-induced high-fat diet animal models and DBDB mice, they develop not only high Phe2 A levels, but also high TLR4. So in the bottom you can see TLR4, and how this happens, how Phe2 A triggers TLR4 is through the phospho-NF-kappa B pathway. That also is clearly demonstrated here. When you have that and look at the serum levels of patients with type 2 diabetes and also mice serum levels, you can see TLR4, then the Phe2 A levels rising, and the Western blot shows TLR4 and phospho-NF-kappa B paralleling each other in these models. Phe2 A causes adipocyte dysfunction. How do you measure adipocyte dysfunction? There are several methods. This is one of the simplest one, known as oil red O experiment. So metabolically healthy adipocytes, they pick up lipids efficiently, and you have those red dots there signifying enough adequate lipid uptake. When you add Phe2 A on the second panel, you can see lipid uptake by the adipocytes. The mouse adipocyte, the human adipocyte, this is our data from our lab. It's a complete abrogation of lipid uptake on addition of Phe2 A. On the third panel, you see rosiglitazone, and you know it is one of the most powerful insulin sensitizers, although no longer in clinical use. So when you add rosiglitazone to the media, naturally the lipid uptake increases in presence of Phe2 A, which is your panel 4. A drug as powerful as rosiglitazone is not able to induce lipid uptake by the inflamed adipocytes. Again, it's completely abrogated. And when the lipid uptake by the adipocytes is dysfunctional, you have those adipocytes releasing pro-inflammatory cytokines. Here you have IL-16, TNF alpha, and IL-1 beta levels increasing. Phe2 A also interferes with GLUT4 mediated glucose uptake in the adipocytes, and insulin, as you know, works by translocating GLUT4 from intracellular vesicles to the cell surface. AS160 is a protein believed to facilitate this process. And Phe2NA is known to suppress this AS160, so it causes GLUT4 to remain dormant within the intracellular vesicles, does not go to the cell surface. Insulin is the most powerful agent to cause translocation of GLUT4 from within the cell to the cell surface. So here you have GLUT4 inside the cell, within the cytoplasm, insulin, you can see how nicely it causes peripheral translocation of GLUT4 the moment you add Phe2NA, even in presence of insulin. Add Phe2NA and all the GLUT4 molecules, they go back into the dormant state within the cell cytoplasm. So powerful is the insulin inhibiting effect of Phe2NA. So this is the toll-like receptor for which you are all familiar with, and it has multiple leucine-rich repeats. As you know, by site-directed mutagenesis, we were able to identify that this is the position 2 and position 6 are actually binding to, responsible for binding of Phe2NA to TLR4. To cut a long story short, free fatty acids coming out of circulation, they stimulate the hepatocytes to produce Phe2NA. We now know that free Phe2NA acts as a binding protein for free fatty acids. So it forms a dimer. And then the dimer goes on to bind to TLR4 present on the surface of adipocytes and immune cells. Then there is a release of pro-inflammatory cytokines that causes insulin resistance. So these are the publications, and we now know that Phe2NA also acts as an adapter protein between adipocytes and free fatty acids and TLR4 to induce insulin resistance. This is the editorial on our paper by Jariolowski, which again recognizes that Phe2NA in fact is the missing link in lipid-induced inflammation and insulin resistance. So again, this is the summary of that cartoon. Phe2NA binding to TLR4, you have release of inflammatory cytokines. Now, how do the inflammatory cytokines induce insulin resistance? There are two proposed mechanisms. One is they enhance the cleavage of SART1 here. SART1, you all know, is good for mitochondrial function. It maintains muscle metabolism, and it's again available over the counter. Now the instructors in the gyms, they also know about SART1. They prescribe, and those gym goers, they buy it from the market and have it to boost their mitochondrial ATP generation. So Phe2NA, by these downstream mechanisms, cleaves SART1. On the other hand, through a series of experiments, we could demonstrate that Phe2NA actually reduces AMPK, and this happens through by interfering with the Wnt3 signaling pathway. So pipa gamma goes down, so does adiponectin and downstream AMPK. So these are two major mechanisms by which Phe2NA finally causes insulin resistance in the adipose tissue and also in the skeletal muscle, and that's your PGC1 alpha. And you all know that how mitochondrial biogenesis and bioenergetics are responsible for ATP generation, adequate mitochondrial function. Both are interfered with due to reduced AMPK and due to reduced SART1. So these are the papers looking at how Phe2NA downregulates adiponectin through the Wnt-pipa gamma pathway, and how impairment of SART1 and AMPK, the energy sensors, they are affected by Phe2NA. I'll go very quickly here. So some Phe2NA suppresses adiponectin gene expression, as you can see clearly here, and when you knock down Phe2NA, you can see the suppressed gene expression is rescued. And the plasma adiponectin level follows. On the GTT, you can see how nicely Phe2NA knockdown leads to amelioration of hyperglycemia in high-fat diet diabetes. this shows the involvement of the Wnt3 pathway by phenytoin A-induced insulin resistance. We have summarized the whole mechanistic pathway of involvement of phenytoin A in insulin resistance in this recent publication. This is from the Journal of IISc Bangalore. Now coming to vanadium. So vanadium compounds were used to treat diabetes since ages. They were the only anti-diabetic agent available before this discovery of insulin. Issues with formulation stability and toxicity were major concerns, and that's why vanadium did not see the light of the day in clinical practice, although over-the-counter use is rampant. Dimethyl peroxy vanadate, to be referred from here on as DMP. So DMP is a non-toxic, water-soluble, oral insulino-mimetic agent that is stable at room temperature, but only for 6 to 8 weeks. Beyond that, it undergoes oxidation. The white powder blackens and loses its efficacy. So that continues to remain a challenge, how to increase the shelf life. That's the crystal structure for DMP, which has been published. So chemistry people may be interested to have a look at it. Now, here is the surface plasmon resonance data. On top, you can see increasing concentration of insulin, showing increasing binding to insulin receptor. So insulin receptor protein, those who are familiar with SPR would know this is fixed on CM5 chip, and so this shows increasing binding, and vanadium parallels binding to insulin receptor. So here is the confirmation that it has insulino-mimetic effect. And you can see the fluorescence spectra telling us the same story. DMP reduced infiltration and polarization of macrophages in the adipose tissue. Here you can see this is hypertrophied adipocyte, big adipocytes in HFD mice. This is a Boyden chamber experiment where you can see the M2 macrophages on top. Once they get polarized to M1, they develop pseudopodia kind of fingers, and they can insinuate through the membrane at the bottom of the Boyden chamber. So the moment you see a diminishing M2 population on top, you know that those macrophages have polarized and they have migrated to the bottom. So HFD mice, here you can see diminishing population of M2, they are replaced with M1, and you have the immunostaining there. You have specific, you all know, you are familiar, there's a specific marker for M1, multiple markers, CD11C being one of them in common use. So marker positive, so M1 with HFD, and how nicely this is rescued by DMP. 50% reduction in macrophage polarization you can see, and this is associated with, you can see phenytoin A level going up, MCP1 going up, so they both are responsible for macrophage polarization in inflamed adipose tissue. At the bottom left you can see this is the stromal vascular fraction of adipose tissue macrophages, and you can see when you have macrophage polarization, you have the inflammatory cytokines going up, and when you add DMP, the cytokine levels are going down because it prevents macrophage polarization. And here you can see anti-inflammatory cytokines going up. The FACS data is very interesting. So this is the 15.6%, you see this CD11C positive macrophages, high fat diet 34%, and this is reducing to 20% on the addition of DMP. So here's the data to show that DMP induces SART1 expression in adipocytes and thereby restores mitochondrial function. Here at the bottom you can see this is standard diet, SART1 content 4.7, high fat diet reducing to 1.6, rescued to 3.1 with DMP. Here you can see macrophage population increasing in the adipose tissue with high fat diet, and this is ameliorated by DMP. This is the aim on positive macrophages increasing and then reducing with DMP, SORT1 activity increasing, TNF alpha going down, IL-6 going down. So on the Wnt3 PPAR gamma pathway, again, DMP actually rescues this pathway, restores the MPK concentration. So here are the studies on mitochondrial biogenesis, bioenergetics, and here you can, on the top, you see how high fat diet reduces mitochondrial density, and this is how nicely this is recovered by DMP. This is the mitochondrial DNA copy number going down with high fat diet, rescued by DMP. Fatty acid oxidation rescued by DMP, and cytochrome C oxidase activity parallels the improvement in fatty acid oxidation, and at the bottom you can see mitochondrial ATP production going up. So here is the evidence to show that DMP induces GLAT4 translocation, and when that happens, you naturally expect your glucose tolerance test to improve, and it does. You can see this is the HFD glucose levels, sky high levels, and there's a more than a 50% reduction with the addition of DMP. And this happens at a lower level of insulin, means DMP is causing an insulin sensitizing effect. So it improves mitochondrial function, it improves pancreatic beta cell function, and it also restores insulin sensitivity. And at the bottom you can see here HOMA-IR going up with HFD and going down with addition of DMP. This is from the mouse epididymal fat pad and on standard diet. So this is the size. This is hypertrophic adipose tissue with a high fat diet, and this is rescued with DMP. Now coming to the insulino-mimetic effects. Here on top you can see with increasing L6 myotubes, increasing concentration of insulin causing increasing insulin receptor autophosphorylation, and with increasing concentration of DMP, DMP also has an insulino-mimetic effect by binding, causing insulin receptor phosphorylation. This happens majorly through a powerful inhibition of phosphotyrosine phosphatase 1B. And then so phospho-IR in the middle you can see that's the Western blot improving with insulin, and at the bottom with DMP, and how nicely both insulin and DMP are causing GLAT4 translocation in L6 myotubes here. So improving skeletal muscle insulin resistance. On a single, following a single oral administration, you can see the half-life is about 5 hours. Duration of action is about 15 hours, allowing for once a day administration. Blood levels are maintained. And here you can see the glucose tolerance. This is a streptozotocin model, streptozotocin mice, insulin deficient, mimicking type 1. And here you can see the insulino-mimetic effect of DMP. So these are models mimicking type 1 diabetes, no insulin in the system. And you add DMP and you see how nicely the blood glucose levels go down, DMP causing insulin receptor autophosphorylation. This is the glucose uptake increasing in the skeletal muscle, fatty acid uptake increasing in the adipose tissue. This is a recent reference to our paper which acknowledges these contributions. DMP is shown to improve insulin sensitivity in diabetic mice. There is a GTT improving, ITT improving, and there is an improvement in HOMA-IR. Detailed toxicity studies have been published. DRDO helped us in doing this. So you have all the standard assays, the liver, the kidney, histology, the hematocrit, the MTT assay, and so on. So these are the publications on vanadium, small insulino-mimetic molecule also improving insulin sensitivity. This is unpublished data on DMP showing binding on reducing the binding to PTP-B1, reducing the levels of PTP-B1 protein levels on Western blot. Coming to the end of the presentation almost. So there are 5 studies using vanadate sulfate. So look at the vanadium content, 31 milligram. In a phase 2 trial, 3 milligram vanadium was safe and effective. This is oxovanadium, but it was not stable at room temperature. Our molecule dimethyl peroxy vanadate, 20 milligram is the dose for a 70 kg adult, 300 microgram per kg. It contains 6 milligram of vanadium. Stable at room temperature for 6 weeks to 8 weeks, no significant toxicity. So if I am running out of time, please tell me. There are 2 or 3 slides maybe. So to summarize, DMP binds to insulin receptor and activates downstream signal transduction, causes GLUT4 translocation, reduces blood glucose levels in both type 1 and type 2, restores energy homeostasis by stimulating mitochondrial biogenesis. It mimics insulin effects in insulin deficient streptozotocin mice model. So I think I have alluded to this before. Metformin vanadium combo producing insulin sensitizing effect. It's the same thing, decavanadate metformin, another chimeric salt of vanadium, improving blood glucose levels. Now with the European Medical Agency and US FDA requiring investigators to outline comprehensive formulation development strategies, this area has become an integral part of the drug development process, which you know better than me. So factors such as composition, dosage form, dosing frequency, methods of administration, specialized labeling and packaging requirements have taken a center stage. So to wrap up, DMP promotes phosphorylation of insulin receptor, tyrosine kinase independent of insulin. So that's the insulino-mimetic effect. It also acts as an insulin sensitizer by upregulating the PPAR gamma adiponectin SAR200 and AMPK pathways. It does not cause hypoglycemia, just like the GLP-1 agonist. It stimulates pancreatic beta cells in a glucose-dependent manner. The moment the glucose levels are below 70 milligrams per deciliter, so DMP no longer stimulates the pancreatic beta cells. So on monotherapy, it does not cause hypoglycemia. It's devoid of major organ toxicity, like I said before. Stable formulation with a reasonable shelf life that remains to be achieved. So that's the challenge. So potential for human trials remains a big question. This is a book that I edited recently, and it may not be a bad idea to have a copy for your library. It's expensive, not selling much in the Indian market, doing well in the Western world, but not here. This is published by Elsevier Global, and thank you for listening.
So now we'll open the floor for question and
Sure.
answer.
So I have a couple of questions. First of all, congratulations on a great body of work, and secondly, it looks very promising, DMP. One is a technical question, that is for type 2 and type 1 models. So what were the durations of treatment that you used?
In animals, 12 to 16 weeks.
Okay, but since the type 1, I mean, if you say this is a insulino-mimetic compound, so in type 1, it should work in a like single dose you can study.
No, this is about type 2 diabetes.
Yeah, in the type
HFD.
1. Yeah,
HFD
what
model.
about the streptozotocin?
Yeah, 2 weeks.
They also, but didn't you see the acute effects? Like if it is an insulino-mimetic, it should work in a single dose actually.
No, after the animals developed type 1 diabetes, I mean insulin deficient diabetes,
Yeah.
it was effective on the same day.
Okay, okay.
That was the same day data.
And second question is, in the conclusion you said like this doesn't cause hypoglycemia, but if it is an insulin mimetic, it might. So what is the difference in mechanism?
Yeah, the most of the DPP4 inhibitors, the GLP ones, they do not cause hypoglycemia on monotherapy, as they stimulate the secretion of insulin from the pancreatic beta cells in a glucose-dependent manner.
Yeah.
A similar finding was observed.
But since you were saying this does bind to the insulin receptor, it's essentially insulino-mimetic in nature. If it does, then GLP-1 is a different story.
That is only part of the story. Its major action is downstream post-receptor inhibition of phosphotyrosine phosphatase 1B.
Okay, thank you.
So I had one question that usually vanadates like sodium orthovanadate. So they are like usually non-specific inhibitors of all the phosphatases. So they will promote phosphorylations of different kind of kinases, like a kinase of interest of you also you got enhanced phosphorylations. So my question was, did you ever check what is the specificity of DMP on the panel of various phosphatases besides PT1V which you are telling?
We haven't done that study. We have just looked at the animal data, toxicity data.
Okay, thank you.
Yeah, very fascinating work. And coincidentally, I met your collaborator, Professor Samir Bhattacharya, almost 20 years back when I was attending one conference in Trivandrum. As you have somewhere, pinpointed the inflammation, which is possibly through MCP, right? Now, MCP, I think, is more systemic, inflammatory sort of indicator or maybe inducer as well. But if you talk about this inflammation in systematic level, that is fine. But if you talk about local levels, like say adipokines or maybe peritoneal regions where the fat is deposited, would that be same or going to be different than what we see in systemic circulation?
The studies that we have done using both Phetuin A and MCP1 were tissue-based studies.
Right.
Here we did not look at systemic inflammation as a whole.
Okay. Thank you.
So the last question by Monica.
So this chimerin and Phetuin A is very interesting. Whether this has come in the clinical practice, it's from the blood or serum? Blood. So it has come to the clinical practice.
No,
people do
yet,
it. Not yet. It can be like very useful to decide the line of treatment. And another thing is like Phetuin A, if you say if it can be blocked, it will not go and block the receptors. So do you think that can be, that kind of molecule can be a solution?
Phetuin A blockers are already available, but at the research level.
Okay. But that can be an interesting
vanadium
solution
as a Phetuin A blocker.
Okay.
Thank you very much, sir. Now I think whosoever has any question can email him directly, because we are running out of time. So we will proceed further. Oh, sorry.
No.
Okay.
Okay.
So time to leave the dais?
Yes, sir.
Thank you. Thank you once again.
Thank you very much, Dr. Satinath, for that deeply thought-provoking and eloquent lecture. We are truly grateful for the clarity and passion with which you presented such a crucial topic. So before we move towards the closing, I would now like to invite Dr. Biswajanani J. Satyagiri to kindly share a few words on how TKDL, the Traditional Knowledge Digital Library, can play a vital role in novel discovery. So, thank you, ma'am.
Namaste to everyone. Thank you, Dr. Radha, for this wonderful opportunity. I think after such a wonderful lecture by Dr. Mukhopadhyay, I do not know what I would add. It actually even, helped me go back to my past. I was with Ranbaxy Pharmaceuticals working on different therapeutic areas, and was also associated with the anti-diabetes drug discovery and development work. It's wonderful to see that, we're still having a lot of active research going on in the academic, while the industry has slowed down so much in drug discovery and development. Well, I think all of you know me. I've been here multiple times and talked about TKDL also multiple times, as you are all aware, and also as Dr. Radha mentioned. The Traditional Knowledge Digital Library, TKDL, is the first of its kind in the world, and that makes me very proud. And we are running in the Silver Jubilee anniversary year. It's almost 25 years since it has been instituted. And it primarily serves the purpose of protecting our traditional knowledge from misappropriation by way of patents. And that is what you would have seen in turmeric, basmati, neem, and many more. Today, the database boasts of about 5.2 lakh formulations, and we have been able to defend misappropriation in about 375 plus patent applications. So when we talk of TKDL, the outcomes largely relate to patent applications being rejected or amended, and sometimes based on the TKDL evidences that we provide, the applications are either withdrawn or abandoned by the applicants. So when we talk of the TKDL, yes, there's so much of promise that it holds, especially for drug discovery and development, because when you look at the drugs that have been approved by the regulatory authorities across the world, almost 40% of the drugs originate with some kind of link to the natural products. They could be natural botanicals themselves, natural products, or their mimetics, or perhaps a hybrid. So somewhere, some connect to the natural products. And when we talk of our traditional systems of medicine, we do have Ayurveda, Siddha, Unani, Sovarikpa, and also yoga practices that help in the health and wellness. In fact, your talk on the vanadium complex, it kind of helped me go back to, the Ayurveda herbo-mineral compositions. In Siddha, it is much more. So mineral component has always been a critical aspect of drug discovery and development. So considering the promise that the TKDL has, the government of India in 2022 has given approval for widening access to the TKDL for all users, and we're still working on the framework. Hopefully, we should be able to wrap it up soon. But when we talk of this, important lecture series, translational research, I think that is where I would like to pitch in to see how is it that the TKDL can be positioned as an important resource for helping drug discovery and development. So on this front, I would like to digress a little bit and reflect back into my past, because you have very aptly pointed out that when we talk of drug discovery and development, it is just so complex. It is not that, one solution fits all. So when I started my career, actually as a postdoc, I got initiated into the medicinal chemistry. I was very fascinated because everything that you do has some kind of, value and application. And that is what medicinal chemistry largely, held promise for. So we worked on this estrogen receptors for breast cancer. I synthesized a few, molecules. We tested. There was always some concern on the selectivity. And then anyway, life goes on. I joined Ranbaxy. In the corporate and in the private sector, what I felt is that while the creativity takes a larger role in the academic and research organizations, in the private sector, it's largely a structured research program. There's a lot of clarity when it comes to what is it that you need to achieve. And, there's the product profile that is well defined. So you know what a hit is, you know what a lead is, you know what a drug candidate is, and so on and so forth. So with this, the empowerment comes in that it's not that, you have everything laid out, you just need to synthesize and put it. The challenges do come, because I'm sure the participants around, the audience around are much more well-versed than I in drug discovery and development. But here, when we talk of, the translational research, it's not just a passion that is important. It is also about the tools that are available to help us achieve the goals. And here I would also like to point out that there are many, many of them. And this I'm referring to in the context of the TKDL being one of the resources to enable drug discovery and development. So when I was a young researcher way back at Ranbaxy, there were a lot of game changers that had come out. A few of them included the Merck Natural Product Library. They felt that this is going to be a game changer. You will have natural products, so many of them, and once you screen them, you will have so many antibiotics. Okay, that was a promise it held. Well, we know what it is today. Then there was this computer-assisted drug discovery CAD or molecular modeling. Yet again, it had a lot of promise. I would not like to criticize any of it because there's always positives and negatives associated with any framework, and that is what even CAD held. When we started with the VLA4 antagonist, we could not, the molecular modeling could not help much because you needed a threefold difference, log difference to, get the the data set right to do the molecular modeling work. But in the case of the DPP4 ACE inhibitor, the molecular modeling did help, and the team that was working on it did derive benefits out of this. Then came the structure-based drug design, especially with the antiretrovirals, and that held great promise. And it did help certain therapeutic areas. Combinatorial chemistry, as medicinal chemistry, I think there was no greater fascination than the combinatorial chemistry. And along with it came the high throughput screening. Then they said medium throughput screening. And yet we are looking at a huge, and a vast unmet medical need. Well, they are game changers for a purpose, and they do still hold promise if we apply the tools right. So when we talk of, especially in the context of drug discovery and development, again, as passionate medicinal chemist, I would be very happy in identifying a lead molecule which fits the bill. But then thereafter came the aspects of, the clinical, doing the clinical trials, making a process that could help doing the clinical trials and so on and so forth. And the foremost and the topmost was what would be the differentiator, the USP for which this molecule can be pitched to take it further. So when we were talking about it, again, the buzzwords or the jargons that were used was first-in-class, backup molecule, follow-on molecules, and so on and so forth. So, and everything relates to only the commercial aspects. Does that make, any candidate, does that make commercial sense when you are planning to take it forward? And then, I moved from Ranbaxy, joined a market consultant group largely associated with the techno-commercial aspects. And when you talk of techno-commercial aspects, it's not just the candidate profile that is important. It is all very important to see how is it going to be positioned in the healthcare, So you had to talk to the key opinion leaders, trying to understand what is it that they want, what is it that a candidate should not have, and so on and so forth. And in addition to all of this, we also had to look at the patent landscape. We also had to, before, to ensure that we are giving the right decision support to the pharmaceutical companies that approached us. We had to do a lot of study and analysis on the pipelines and also the clinical trials. I think that was the largest and the most difficult aspect, because not much of information is put into public domain when you consider molecules which are in development. But trying to get that information and then help your clients to understand whether it is good to go with that particular therapeutic area or a candidate was much more important. So there was a lot of struggle. So when we talk of translational research, these are all the aspects. It's not just that, we take a molecule that has been synthesized in the laboratory and then, project it for clinical trials. That is not translational research. In the domain of drug discovery and development, they are much, much more complex things that one need to worry about. And when we, in today's terminology, is considered translational research, again, a very important aspect is to understand the TRL levels, that is the technology readiness levels. And you actually get into the domain of drug discovery and development, get into something meaningful only when you pass the TRL 4 level. That is when you establish the POC from both efficacy and as well as the safety parameters. And your clinical trials is still far away. You get into, once you do the phase 1 trials, that is when you actually enter the TRL 6. Unlike any other engineering technology, that, it is easy to develop a prototype and and show, yes, I have something great. So here, when we are talking of translational research, I would again like to go back again to my Ranbaxy days and refer to the importance of knowledge networks. As I think you also mentioned, and Dr. Radha also mentioned about collaboration. Drug discovery and development is a very complex subject matter, and collaborative networks are indeed very critical. In fact, Dr. Radha and I, we were appreciating your collaboration with NII and also IICB and so on and so forth. And I think this is also very critical. So I would like to quote Jean-Paul Garnier. He was a former CEO of GSK, and at that point of time, Ranbaxy went into a collaboration with GSK to develop molecules for COPD and some other diseases. He said, for solving productivity problems, one needs to morph big into the small. So what does he mean? R&D needs to be reorganized into small, highly focused groups. He then goes on to say, companies need to seek the best science wherever it resides, inside or outside the company. So this he mentioned in the context of the importance of knowledge networks and as well as markets. So coming back to this point of knowledge networks, in the context of even Ranbaxy, we got into collaborations, as I mentioned, with the GSK. We also connected with the CDRI. Gugulu was taken up, okay? And this was for the anti-diabetes treatment. So coming back to the main reason why I've been invited to talk about the TKDL and, connecting it with the Gugulu experience at Ranbaxy, I'd like to mention that TKDL, yes, it is an important source because you have 5.2 lakh formulations. Perhaps it is one of the largest repositories of traditional knowledge across the world. But there is much more, when we talk of frameworks to help doing this translational research, the national IPR policy, the Bio E3 policy today, they are all very facilitative in the context of helping us achieve our goals, especially related to addressing unmet medical needs. I'd like to kind of mention as to why herbal medicines are important. A recent analysis of the patents that have been filed during the COVID-19 period, we are talking just about 2020 to 2022, in a 3-year period, almost 4,900, I would say 5,000 patent applications were published as therapeutics, and almost 10% of these applications, 464 applications, were relating to traditional medicine therapeutics. And out of this 464, 59 came from India. And most of these applications relate to herbal medicines as formulations for boosting immunity and or as antivirals. So it does hold promise. Even in times of calamity such as COVID-19, herbal medicines did hold importance, and it will continue to hold importance. When we talk of healthcare, we need to realize drug discovery and development is not the only framework to help the healthcare needs. In the context of the TKDL herbal formulations and as well as healthcare, I'd like to mention that the TKDL does have a lot of potential for helping address the healthcare needs as foods, as functional foods, as dietary supplements, as herbal products, and as well as phytopharmaceuticals. In this context, I would like to mention that yesterday I received a message. Johnson & Johnson has brought out a formulation of turmeric and as well as tamarind for help improving immunity and as well as the bone health. Okay, and this is already being sold in the market. So when companies such as Johnson & Johnson can do that, I think India can do better. And I would look forward to having you all support the TKDL, and I'm there to support all of you in your endeavors related to drug discovery and development and helping achieve the societal needs on healthcare. Thank you so much.
Thank you, Dr. Bisajanani, for your insightful perspective and highlighting the importance of integrating traditional knowledge with modern scientific practices. To mark our appreciation, now I request Director CDRI to kindly present a memento to our speaker. Dr. Satinath, please. सबको बुलाना. Okay. Dr. Viswajanani also. Please come. Thank you, ma'am. Now, to formally conclude the event, I would like to request Dr. Arun Trivedi to deliver the vote of thanks.
So now, good afternoon, everyone, or rather good evening now, I guess. On behalf of CSIR-CDRI and members of the Translational Research Lecture Series Committee, I extend my heartfelt thanks to our esteemed speaker, Dr. Mukhopadhyay, for delivering such an insightful and thought-provoking lecture. I would also like to thank Dr. Satigiri for accepting our invitation to grace this occasion as a chief guest on a very short notice. I also thank Dr. Radha Rangarajan, director of our institute, for all her support in the successful organization of this event. I would also like to thank all the faculty members and staff involved in organizing this event. Special thanks to Dr. Rajdeep, Dr. Vaisakhi, Dr. Vivek, Dr. Sanjeev Yadav, Dr. Anand Kulkarni and their support staff for ensuring smooth and successful organization of this event. A very big thanks to all the students and participants for their enthusiastic involvement and attentive presence. Your engagement made this session interactive and very lively. Once again, I thank you everyone. Thank you very much. Thank you.